We're good to go.
Okay. Good morning, everyone. I'm Cynthia Carnes, and I'm happy to invite you to this session on managing research integrity during the COVID-19 emergency. This is sponsored by the OSU Center for Clinical and Translational Science. And as we went into the research pause and we're thinking about how to support the research community at Ohio State and at Nationwide Children's, our thinking was that it would be good. This is a nice time to sort of, during the pause, to stop and think about processes, process management, and things we can be doing, even working remotely to improve our research programs. So we've got, I think, a really nice set of speakers lined up. We will start with Angela Tsao. Angela actually is known to us at the CCTS. She is the Hub Liaison Team Administrator for the Trial Innovation Network at Ohio State. And the Trial Innovation Network is a collaborative among all the CTSA institutions across the U.S. at academic medical centers and is there to enhance multi-site trial operations. Angela will be speaking to us today about thoughtful data management using standard operating procedures, or SOPs. Angela, I'm going to turn it over to you.
Okay, can everyone see my screen and hear me okay?
Yes.
Great, okay. So thank you, Cynthia, for the nice introduction. Good morning, everyone. Just as Cynthia said, I am a research manager in the CCTS, in which I manage two programs, the Trial Innovation Network program, as well as our Participant Clinical Interactions program. I am also a member of the College of Medicine Clinical Trials Management Organization, in which I serve as a project manager for large multi-center research studies. And lastly, I'm an instructor in the College of Nursing for a graduate course in project management for healthcare and clinical research. Today, the outline for this presentation includes a discussion of COVID-19 implications for research data management, a look at federal and local research guidance, as well as local resources, essential SOPs, and then just a look at an application of COVID-19 related data management practices in a pragmatic multi-center randomized controlled trial. So as you know, the COVID-19 pandemic has disrupted healthcare delivery systems around the world. Clinical and translational research projects are not immune to this massive disruption. This global crisis has had significant impact on clinical trials for current and future study participants, sponsors, investigators, coordinators, and regulators. Thoughtful consideration on how to manage clinical trials during the COVID-19 pandemic is of particular relevance to the studies with study populations that were defined as vulnerable populations pre-COVID-19 pandemic. Additionally, there has been a rapid reclassification of what seems to be a vulnerable study population in an overnight transformation due to the fact that many people are high risk for COVID-19 related morbidity and mortality. With the implementation of social distancing restrictions across the globe, barriers have been erected both literally and figuratively that has presented substantial challenges in adhering to research data management best practices. So where do we go from here? How do we make sense of the current turbulent environment? The answer is to find and harness calmness within the storm that is the COVID-19 pandemic. And you may ask, what is said calmness? It is a combination of staying apprised of current federal and local guidance and developing adaptive standard operating procedures as swiftly as possible, using federal, local, and institutional guidelines to inform the development of unique data management SOPs. And these can be used as your umbrella amidst the storm. So to start with the big and work our way in, we can familiarize ourselves with the vast federal guidance that has been released in response to the COVID-19 pandemic. On March 9th, the Executive Office of the President's Office of Management and Budget issued umbrella guidance for recipients of federal funded research grants. The OMB guidance may have enabled awardees to breathe a small sigh of relief as the federal government acknowledged the inescapable ensuing delays to ongoing research and granted extensions on submission of financial and progress reports, as well as carryover balances. The US FDA, NIH, and NSF, among numerous federal agencies, have provided guidance on the conduct of clinical trials during the COVID-19 pandemic. The NIH and NSF guidance expands on the OMB memorandum and outlines the flexibilities available to recipients conducting NIH-funded and NSF-funded clinical trials in human subject studies that are impacted by the declared public health emergency for COVID-19. The FDA guidance provides general considerations to assist sponsors in assuring the safety of trial participants, maintaining compliance with good clinical practice or GCP, and minimizing risk to trial integrity during the COVID-19 pandemic. This guidance was first issued March 18th, but has been routinely updated, with the most recent update occurring on May 11th. The Office of Human Research Protections, or guidance, provides a set of recommendations for research in the setting of the COVID-19 pandemic. Notably, ORP highlighted the need for investigators to eliminate immediate hazards to research participants prior to IRB review and approval. However, changes need to be tracked and reported to the IRB when possible. As an additional resource, OSU has compiled a listing of over a dozen federal agencies' guidance at the website listed here. Locally, our institution has been issuing guidance for clinical and translational research frequently. On March 12th, the Ohio State Office of Responsible Research Practices released guidance pertaining to human subjects research amid the pandemic that is applicable for all human subjects research conducted at Ohio State, regardless of the reviewing IRB. This guidance defines essential versus non-essential research and provides and specifies requirements for being able to enact changes to the study protocol in lieu of IRB approval. However, changes should be limited and will need to be submitted as an amendment or protocol deviation in accordance with the study IRB's procedures for submission of amendments and deviations. Dr. Stone's guidance for all research and creative expression investigators emphasized the need to prioritize critical research and recommended the development of contingency plans. So despite the fact that we are all confronted with unprecedented challenges in the wake of the COVID-19 pandemic, I believe it is important that we don't throw out the baby with the bathwater whenever possible. Years ago, the College of Medicine Clinical Trials Management Organization developed a set of 20 clinical research SOPs covering fundamental aspects of implementation of clinical research. And while the CTMO SOPs are geared towards FDA-regulated research, these SOPs are adaptable and customizable to the unique needs of many different types of research teams. Two SOPs in particular that are relevant to our discussion today regarding managing research integrity include the SOP on writing, training, and maintenance of SOPs, and the SOP on data management. These SOPs have useful templates, including a template for SOPs and research procedure addendums. The CTMO SOPs are located behind the firewall on OneSource, but they're also more broadly available through the CCTS website. And additionally, the OSU Research Commons previously developed a series of best practices for research data management. These are also publicly available on their website. Lastly, it is important to stress that the basic principles of using SOPs apply to all research, both COVID-19 related and non-COVID-19 research. It is my hope that pre-COVID-19 pandemic studies teams were already utilizing SOPs in some capacity in the implementation of their research. However, all study teams have been forced to confront unanticipated conditions, and therefore there could be a need to develop new SOPs or expand upon existing ones. There are some additional electronic tools for research data management available to faculty and staff, including REDCap and Encore. Listed here, REDCap is it's a CFR Part 11 compliant and HIPAA compliant secure electronic data capture software that can be used for building and managing clinical and translational research study databases. And it has the capability to be able to capture the electronic informed consents. Encore is a secure web-based clinical trials management system that captures all aspects of the clinical trial lifecycle. The College of Medicine Clinical Trials Office, or the CTO, oversees the facilitation for oncology trials, use of Encore, and then the College of Medicine Office of Research oversees non-oncology. Groups like the CTMO and CTO have been diligently and systematically updating research records housed in Encore to reflect the status and changes to studies in response to COVID-19. So it is important to be familiar with the various resources available to study teams, and equally so, it is important to be mindful of the inherent responsibility of investigators and study teams in being good stewards of data and information at this time. The safety and well-being of study participants and research team members are of utmost importance. Actions that enhance the safety and well-being of study participants are consistent with the core research principles of the Belmont Report, including beneficence, justice, and respect of persons. It is everyone's responsibility to uphold these principles in managing research data and information. The development and implementation of SOPs is critical to ensuring quality data and reducing risk. And SOPs in response to the COVID-19 pandemic are needed for various risk mitigation efforts in order to ensure reliability of the data that will subsequently be used to make scientific conclusions. Detailed SOPs should be developed and provided to investigators and coordinators. SOPs listed here regarding the rise of telehealth and remote assessments. Also, if the study is not paused and is continuing enrollment efforts remotely, there should be SOPs related to the electronic consenting and new enrollment process. Also, there should be an outline documenting how out-of-window follow-up visits and just the cessation of any non-essential study procedures or interventions, and adverse event reporting and specific issues regarding adjudication definitions in light of COVID-19 infection should be adjusted in if there is a clinical events committee SOP or manual of operation. Additionally, statistical considerations should account for possible asymmetric enrollment by geography, considering that across the US landscape, various regions are open and closed, so would want to come up with a way to document this, and so that may mean revising one's statistical analysis plan. Also figure out a way to account for results before and after the pre-specified date in which COVID-19 had a significant influence on trial conduct. Statistical monitoring methods should be considered that can remotely evaluate trial safety and conduct. And consideration should be made to assessing COVID-19 serology if biospecimens are available in order to conduct an exploratory analysis stratified by COVID-19 exposure. So these are all elements that you would want to revise and capture in one's statistical analysis plan. Recruitment plans may need to be revised to reflect new recruitment and screening practices. And as you know, DUAs or data use agreements often grant research access to a wide array of protected health information. Data management plans outline how data will be managed and documented throughout a research project, inclusive of the appropriate security and privacy controls. for complying with data use agreements. These agreements and plans may need to be updated to reflect new or heightened safeguards necessary for storage and transfer of data and other files. And then also one's SOP for how to report protocol deviations may need to be updated as well. And so here, just very briefly, I will just talk based off my own personal experience for a trial in which I do serve as the project manager. It's the LOFT-HF study. This is a study in which Dr. Abraham, cardiologist, is the PI here at Ohio State. It's a study evaluating the use of oxygen therapy for the treatment of central sleep apnea in patients with heart failure. This is a large multi-center trial. Right now, we currently have 25 sites with plan to scaling up to 50. The study has central IRB oversight through Vanderbilt University, and we do have a comprehensive manual of procedures. And so in response to the COVID-19 related data management practices, we issued a memorandum, first a study-wide memorandum of cessation of in-person enrollment, follow-up visits. We've done at least 2 surveys in which we just tried to get a snapshot of where of the conditions at each of our sites, because they are spread out across the US. We have frequent site communications, trying to keep everybody engaged, and we have already amended the protocol once to expand on inclusion criteria that would allow us to capture patients based off of previous clinical procedures, instead of doing I guess we open up 2 routes. You could either have a previous clinical procedure, or there's still the permission to, or the ability to enroll based off of doing a study specific test to determine if one has sleep apnea. And so for us, with our manuals of procedures, that is our collection of our SOPs. We've leveraged the involvement of many of our different committees to help us revise the various procedures listed here. And we do also plan to amend the protocol once more, just to allow to actually incorporate the electronic consenting into the protocol. Our IRB of record has said that temporarily this is allowed and would need to be reported as a minor protocol deviation at the time of a continuing review. However, if we do plan to do this long term, it does need to be, captured in the protocol. And so we are anticipating that, this crisis to last for some time. So we're going to amend the protocol accordingly. Lastly, I'll just say if anyone needs any help or has any questions regarding research SOPs, feel free to email me. And that is it. I'm right at 9:20.
Thank you, Angela. I will point out that Stuart has pointed out to us that if you have any questions or comments, you can put those into the chat. I see a few things coming in about the university research data policy. Stuart, we should definitely capture the comments here in the chat and make sure that we get that all put together. There's also a comment that REDCap is not quite 21 CFR 11 compliant, but should be by August of 2020.
Okay.
Okay.
Thank you.
Thank you so much, Angela. That was great. Next up, we've got Dr. Abigail Norris-Turner. While she's coming on here with her slides. Hi, Abby. I'll do a quick introduction. Dr. Norris-Turner is Associate Professor, Department of Internal Medicine, and her research focuses on women's reproductive health in a global context. And the reason we have her here today, and she's spoken for us on this before at the CCTS, but she has served on the university committee that developed policies and guidelines for authorship agreements. So this next segment is really about authorship and collaboration agreements together. So I will turn it over now to Dr. Norris-Turner.
Thanks, Cynthia. I am realizing that I'm not sure how to show my slides in the display in the presentation mode while I'm sharing with you. Let me see if I can. There we go. Does that look as it's supposed to on your side?
Yeah,
okay, great.
that looks good.
Fantastic. So thanks so much for that introduction, and really it's great to be able to come and speak with this group about this important topic, authorship agreements, and what they look like under normal circumstances, and what the considerations are when we're thinking about how to structure this essential part of the work that we do, which is the dissemination aspect. So how do we do it under normal circumstances, and what might go wrong, and what are some best practices about that? And then I want to spend, of course, some time talking about how that looks different in the current situation. I note that the title of this seminar, as it was developed a few weeks ago, is the COVID-19 emergency. And I don't mean to dismiss that it is an emergency. It is an emergency, but my strong feeling about what it means for our work is that we can no longer continue on with our research and with our collaboration agreements as though we're in a very limited time period where we can kind of string things along with changing all of the rules and expecting to go back to normal at the end. We really have to think of this as a shift much more broadly, in my opinion, and begin to make sure that we have strong systems in place, because we're in this for a bit of time. Anyway, I'm sure we'll have time to get into that. So here's a brief outline of what I'm going to go over. I'll talk quickly about the history and the purpose of authorship agreements, including guidelines that Ohio State has developed over the last couple of years, and some best practices. As I mentioned, I'll talk about the problems that arise with authorship agreements in particular, and sort of how COVID exacerbates and complicates some of the problems that exist all the time, but maybe make the mitigation of those problems more challenging. And I just want to call out something that I have heard over and over and over in the last period of time, and that's the advice that you may have heard from your mentor, from your division director, department chair, dissertation advisor, depending on who's listening, that it's a great time to work on your papers. You can't continue your field work maybe if it's with human subjects, but it's a great time to buckle down and get some work done. And I would just put out there that this is really true for some people, that it's a great time to work on your papers. And it's really not true for other people. For some people, the current period of time is actually a terrible time for them to be trying to work on papers. And so part of our system related to authorship agreements in this crisis has to account for the fact that some people actually cannot be productive at all in this period of time, and trying to figure out how we're going to make space for that, and continue the development, particularly of junior scholars who are really disadvantaged by potentially personal life colliding with professional life, colliding with career stage. And authorship and papers is maybe one of the most visible places where we're going to begin to see manifestation of those disadvantages and vulnerabilities. I should also say I'm really interested. This kind of topic is much richer when it's informed by audience feedback. So as Cynthia mentioned, I spoke about this a few months ago, and I think I talked about 10 minutes, and then we had a group discussion for much longer than that. So I'm going to go through some basic ideas here, but I would really welcome feedback from all of you about questions or discussion points that we can talk more about. So authorship guidelines in general is a set of principles that establishes how authors are added to a piece of work. So the type of work, of course, is going to vary across fields, and there will be discipline-specific differences about who traditionally gets put on papers and who is not. And authorship guidelines broadly don't try to dictate or prescribe what different disciplines should do. There's a kind of broad agreement that there's going to be variability there. But there are some common principles and protections that can be made more generalizable across many areas. And so OSU's project over the last couple of years, starting in the University Research Committee, and then kind of extending out broadly, was to formulate and sort of codify what some of those broader guidelines are, and create a set of guidelines. It's not binding. It was an interdisciplinary process, meaning there has to be some allowance for those differences across groups, but it's a set of guidelines that is meant to start discussions between potential authors around formation of that eventual list that goes at the top of the paper. So I'll share at the end of this, the agreement or the guideline document, which at the end has something called the author contributions and disclosure worksheet, which sort of says who did what for the project. It's a valuable instrument, and a lot of work was put into making it. I want to stress strongly that, like so many things in research, what the document and the guidelines do is prompt a research team to have an open and informed discussion about expectations, ideally before a lot of the work is done, and really importantly before the paper itself is drafted, that explains what the responsibilities are of different people who are part of the project and what their reasonable expectations can be related to authorship. And so the worksheet, even though, as I say, it's not binding, what it really does is guide teams through the process and potentially and hopefully through the negotiation. If there are two people who think that they should both be first author, they have that conversation before there's a dispute, once they've both already made the contributions that they think lead to their each being first author. And then the guidelines also outline a process for authorship disputes, which, depending on who's having the dispute, kind of go through different mechanisms. So including the best practices, just to tick through them, and many of you already know these. So best practices for authorship, of course, requires accurate disclosure of conflicts of interest. We have a revised conflict of interest disclosure process at the university right now, which these things all work together, right? In the global project of improving research integrity is disclosing those things. Of course, the funding, and it has some other best practices, for example, use of the ORCID and other collaboration agreements that sort of display for different disciplines, kind of what are some other tools that might be used to work out authorship. The guidelines also talk about unacceptable authorship practices. So you've heard these terms before, the 3 Gs. So guest authorship means having someone participate on the work who didn't do anything related to it, but just to honor them or to provide credibility. So, assuming that the person your paper is more likely to be accepted if an expert in the field is kind of added to the paper. Gift authorship. So this is sometimes someone you like, someone who needs a paper for their CV, someone who maybe worked hard in a different area, but really didn't contribute to this paper, but so putting them on the paper as a reward. And then ghost authorship is when someone really does do a lot of work, but they don't get named as an author. And this is a different kind of crisis related to credit and disclosure. This is usually when the paper's been authored by a professional writer or potentially even someone who sponsored the work, but people perceive correctly that would be considered a conflict, and so the person is not named. And so the guidelines talk about these things. I won't spend too much time going through these, except just to say that this idea of authorship guidelines is really important to a lot of different organizations within science because it takes up so much time when there is a dispute, and it's really can be such a professional crisis for people who feel that they're not getting credit for work that they did, and is really disruptive to their career path and to the progress of a lab or the productivity of a lab. And so, NIH is really interested in having PIs, sort of senior author type folks, really lead discussions amongst junior lab members or team members or graduate students to kind of write out these things ahead of time with expectations. So there are no surprises, or as few as can be managed, and kind of working out those disagreements ahead of time. So I put this up here mostly to show the kinds of questions NIH would like each of us to be thinking about, but also you know, if you Googled authorship guidelines, you would find every university has something about this and something out there. And so there are many. I like these NIH ones, but there are others that may work better for different types of research. So that was the first page. Here's the second page. You can see NIH has quite a lot to say about these things. So what goes wrong in the sort of normal times with authorship disputes? And here I've already alluded to the fact that public health, that there's field or discipline variations. The one that I like to point out is that the disciplines of public health, I'm an epidemiologist and medicine, and I'm in the division of internal medicine. The authorship expectations related to effort and where you end up on the list are actually different, even though those are quite related disciplines. So in public health, the order of authors generally, not 100%, but generally is the order of effort on the project. We don't have nearly the same degree of commitment to the senior author, the last author being the senior author, and that being a prized position. And in some areas of public health, the last author is the one who did the least amount of work. So you can imagine that for people who are working across disciplines, there would be expectations of who sits where that can really cause conflict if you don't call those out ahead of time. I do a lot of work internationally, and the questions around international collaborations and authorship are not new and are not trivial. And this relates in some ways to things I've already talked about related to just discipline differences, but there are other ways in which international collaborations really rely on huge amounts of efforts from folks in the field without whom the research couldn't happen, but whose contributions aren't on the list that journals normally recognize as indicative of authorship. So field staff, but also more senior members of established collaborations who have to do, for example, have to negotiate on your behalf with the Ministry of Health in order for your work to go for approval to the foreign institution's IRB. Like, you couldn't do the work without that person, but that isn't like a named kind of authorship role. So how do you talk about those things with authorship? And then the last category is program grants. So of course, a PI over a whole program doesn't have time to contribute to individual papers in the same way that they would if they were just the team lead of one or just the PI of one project. But they've done a lot of negotiation to get the whole thing fielded. And so what can their expectations be to be on all the papers from their program grant? So pivoting a little bit to talk about the situation with COVID, I'm just going to remind all of us that what you know, because you're living it, is that this period, starting about 8 weeks ago, has just been hugely disruptive for people, just like profoundly changing the ability for people to be productive. So that relates very directly to authorship. There may have been authorship agreements made that relate to timelines and figures that are supposed to be produced, experiments that were meant to be finished, things that were meant to be drafted and sent out. And some people on the research team are not going to be able to do those things. They may be caring for unwell relatives. They may be sick themselves. They may have kids at home that they are homeschooling. They certainly have the kind of emotional burden that we're all managing with this great amount of worry that we have around what's happening in the world. And I should say, I'm an infectious diseases epidemiologist, so I'm projecting here a bit, but I feel that this is a common experience of the academics around me. We don't have the ability to control our schedules in the way that we did 3 months ago. And some of us, and I'll put myself in this category, have been pulled off of the work that we had been doing and have really been shoulder to the wheel on other efforts on behalf of other institutional partners, the city, the state. And so there's just, in my own space, there's no way I'm going to meet the expectations that I had a few months ago. And these issues require us to be more flexible with things that people are producing, and especially to people with existing, as I say, vulnerabilities and disadvantages due to position and career stage. So I'm very grateful that since my existing work has stopped and I've been pulled 100% into helping with the state response to coronavirus, that I already am a tenured associate professor. I have this incredible stability in my position. That means that I'm massively failing to meet expectations, but I'm okay. And that is not true for many people who feel very, very worried about the things that they're not producing. So, you know, and here's just one example related to authorship. So what happens when critical work can't be done in a timely way by one team member for some of the reasons I've just described, but another person who is in a different financial, personal, et cetera, position, if that person can do the work, right? So on the one hand, that's fantastic. The broader project can move forward because someone else can step in. But what if the first team member, who was meant to be the first author on the paper, needs that paper in their dossier in order to be put up for promotion or to be nominated for an award, or even just to have a positive review? How do we acknowledge work that was done up until that point, and also give credit to the person who was able to step in? And all of these things require you know, a large degree of communication with the team members. So kind of like Angela was saying about SOPs, revisiting an existing agreement is much easier than starting a difficult conversation when we're already in a crisis. I realize that we're already in the crisis, so that advice can ring a little hollow. So let me just follow it up by saying having an agreement or starting a conversation right now while the work is paused is also easier than having that conversation 2 months from now, when all of the kind of differences across productivity might become more obvious and the expectations might be more challenging to describe. So I've put some references here related to further reading, and I think Stuart can distribute these slides out to folks after the presentation. I'm gonna stop for a second and just pull up the OSU authorship guidelines and show you that worksheet that I was describing. And we can also share these out with others. Ooh, here we go. So this document, it has a whole set of guidelines here. Some of the things I've shared with you around best practices, but the part I really wanted to draw your attention to here is the author and contribution disclosure statement. And this is the framework that people can use when they're going through to talk about what their roles were on the project. These guidelines were developed actually to generate discussion amongst teams, but also to have a kind of a record of who did what with a given paper, so that if there's any question later on about, you know, research results or, you know, what was presented in a given figure, et cetera, there's some written documentation that, like, this person was responsible for figure 2 or table 1 or whatever. But as I say, it's really just a conversation generating tool. So I think I'll stop there. I know I have 10 minutes left. I hope to be able to answer questions for people if you have any. Yeah, thank you.
Okay, thank you so much, Abby. I think what we'll do is I'll go ahead and talk about collaboration agreements, and we'll see if questions come in, and then we can maybe through the chat, we can address those together.
Great.
Then. There we go. Good morning. I'm Cynthia Carnes, and your MC and host for the event in collaboration with Stuart Hobbs. I'll be giving a brief overview on collaboration agreements, which is something that I think about a lot in my role as Senior Associate Dean for Research at the College of Pharmacy, also as the College Research Officer, and as the Director of the KL2 program, which is a junior faculty training program run through the CCTS. And this is an interesting time to think about research collaborations, and I'll try to elaborate on that as we go through the material. So, you know, a frequent phrase that you will hear when we talk about thinking about collaboration is development of a scientific prenuptial agreement. Or what I like to think of it as hoping for the best while planning for the worst. So really thinking about this in a prospective manner is absolutely critical. And as I've been thinking about this during our time of working from home, I do think that due to the differences in communication and communication styles, I think thinking about doing this more formally becomes even more important.
We seem to be experiencing some technical difficulties. Please stand by. This might be a time to ask a question, Allison, while Abby, I'm sorry, while Cynthia may determine that she may have to re-log in or something since she is frozen out. What other comments would you like to make, Abby, while we're waiting for Cynthia to come back again?
I was just looking up the guidelines to be able to share them in the chat window. I see that Susan shared the link to them, and I was going to share the actual document. Cynthia, are you back?
I think she's come
am back.
Great.
So the ironies of talking about the difficulties in communication and then having it zoom out.
Very nice practical demonstration. Thank you.
Yes, it really was. I was going to say, after all these weeks of being at home, that's the first time I've dropped in the middle of the Zoom, wouldn't you know?
You just have to hang with the technology. Okay.
That's it. So let's just move ahead with key elements of a collaboration plan. So you want to be as specific as possible. Who's doing it? And I think if you have trainees or staff involved, you want to be really, really explicit about expectations. so that that's extremely, extremely clear. Because I always think that when there's a collaboration, they are the most vulnerable to disruption.
But if you could share your screen, Cynthia. You can do that while talking.
Yes. It says the host has disabled participant screen sharing. You're going to have to re-host me, Stuart.
Carry on and I'll get you
okay, sounds good. I think the other thing that one would want to consider is thinking about organizational structure. So things, and this really dovetails very nicely with what Abigail was just discussing, is that our, you know, I think time and effort expectations may be different right now than they are during normal times. Are we back?
You are back.
Okay, excellent. So I think the things that I would say is, you know, time and effort expectations do have to be varied right now. I would say sometimes we have the best of plans and things happen that make it more challenging for us to stay on target. I think setting up meetings and communication for frequency, who's calling the meetings, I think is really important. Sometimes what I find is that nobody's calling the meetings and they're not happening. So setting expectations for those, even if it's just I need to reschedule during this pause, I think becomes incredibly important. One of the most important things to think about at the outset of a collaboration is regulatory responsibilities as you're dividing the work. So if you've got compliance issues around animal research, IRB, whatever the regulatory issues are, data management, who's going to take charge of that? Who's going to be responsible? And getting that written down so that it's very clear becomes very important. Other things that I think are really important to talk about, and Abigail touched, of course, on authorship, which is a highly sensitive matter, because I always say, you know, publications are our academic currency, so those can be a real flashpoint. Compensation and financial management. Who is going to be responsible for managing budgets, making budgetary and spend decisions? That really should be laid out at the beginning of a collaboration. I definitely encourage people to think about subawards as a way to keep things clear instead of having one big pooled budget. That's something that certainly can be considered as a way to maintain sort of clarity and manage expectations. Other really important things to think about are data sharing. Who owns the data? Who gets to see the data? Is everybody getting to access it when they want it? Who's responsible for managing and organizing it? And then who's responsible for retention of the data? All these things. And then intellectual property agreements become incredibly important when we start collaborations. Whether those are within an institution, which simplifies things if we're all at Ohio State, the intellectual property is governed, you know, through one office. That becomes very simple. If we get into multi-institution collaborations that are generating intellectual property, we'll need to really think that through prospectively. We may need to consult the technology commercialization office at each institution and get that figured out. Again, academic and non-academic institutions have very different approaches to intellectual property. So really taking some time and asking yourself, is it possible that there will be intellectual property generated through this collaboration? And thinking about that beforehand is absolutely critical. Another thing that can happen if you don't think about it carefully is thinking about who gets to own, keep, and control customized reagents, constructs, mice, all those different specialized research reagents, and who maintains those and is responsible for those. Another issue that can come up is disposition of the reagents after the collaboration ends. Certainly for federally funded research, there's an expectation that we would be sharing our research resources, but what if it's not federally funded? What if your collaborator generates specialized constructs? Do you get unlimited access to those, and for how long? all these things really need to be thought through. What is the scope of use? If I want to move on from our collaboration and do something completely different with the reagent, is that okay or not okay? Do I need to have permission from my collaborator beforehand? Again, I think all these things are really important to think about ahead of time. The other point I want to make, and again, this gets back to the idea of a prenup. I would say expect the best, prepare for the worst. You want to be planning ahead of time for conflict resolution. Hopefully if you have everything written down, you're not going to have conflicts. But if for some reason you don't contemplate everything ahead of time, or things don't go the way you think they should, who is going to mediate a conflict between collaborators? So often people will designate department chairs, college research officers, etc. Certainly, you could have somebody in the Office of Research be sort of the top level mediator after you go through different steps of mediation. Somebody needs to be designated to have the final decision-making authority. You can imagine if it's a multi-institution collaboration, this can become even more complex. I wanted to point out, and I think Abigail had the same slide put up, but it's really important to think about all the steps. There's some really nice guidance from the Office of Research Integrity. Again, it sort of spells out all the steps that one would want to take. And I guess as I've been thinking about the pause, the thing that I would say that I think is incredibly important is that normally I think when things are not going well or you have a collaboration, lots of times, I think about just going down the hall or picking up the phone. I think communication takes a little bit more planning right now while we're all working from home. So I would just encourage everyone to take this time out to really think about how they're managing existing collaborations, but also as we're at this point of thinking about new projects, new grants, what is that going to look like for the future, assuming that we don't know what a return to the new normal is going to look like or when. Thank you for your attention. I'll stop there. So we've got a few questions in the chat. Anything else, Stuart, that I missed question-wise? Let's see. And just as a reminder, it does look like Stuart will be sending out the slides and materials after the talk as well. Okay, next up, and it looks like we are pretty much on schedule. Next up would be Dr. Phil Binkley, who's been assigned the really interesting topic of mentoring in crisis times, very timely. Dr. Binkley is a cardiologist and director of the Center for Faculty Advancement, Mentoring and Engagement, or FAME, and is a professor in the College of Medicine and is a nationally known authority on mentoring and faculty development. I'm going to turn it over to Phil. Thank you.
Thank you so much, Cynthia, and it's good to be with all of you today, even virtually. In full disclosure, I am at home and I am about 12 feet from my two Sheltie puppies. So you may at times hear them contributing their thoughts on mentorship as well. So please forgive me for that. So, it says host is disabled participant screen sharing. So is it possible, Stuart, for you to allow me to share my screen?
Yes, it is.
Okay, good. Let's try it again here. It still says disabled, so I'll wait another couple seconds. Okay, there it looks. Thank you, Stuart. Looks like we're ready to go. So first, let me tell you what is informing a lot of my discussion about mentorship in general. And I know we have a lot of accomplished mentors in the program too, so I would also love to hear their thoughts and advice also. But the comments here are based on in part a mentorship best practices document that Stuart is making available to you that was actually created by focus groups in the CCTS as well as the College of Medicine and is our official mentorship best practices document. My own experience in mentorship as well as hearing from students and other mentors. I teach a course and co-direct an MPH in Clinical Translational Science. And in my courses, I hear a lot from developing scientists about some of their successes as also well as their challenges in mentorship, and can share some of those with you. I think another amazing resource that many of you may have access to or have seen is this document. It is a consensus study report of the National Academies of Sciences, Engineering, and Medicine about mentorship. It is an amazing document that covers so many aspects of mentorship based on listening groups and also literature review. I've read, it's a very long document, cover to cover, but I think you'd find it's also a useful resource to simply pick up on certain topic areas that...
We're still not seeing your slides, Phil.
That's interesting. Well, let's see.
I hope I've got you set up right.
Okay, let me try this again. Okay, let's see.
How would you be
okay,
logged
maybe
in?
Here now. Let's see.
There we go.
Good. Okay, good. Well, so I won't go back and repeat those because you really miss a lot. Except let me start the slideshow so you can see the slideshow format. And I think what is important is if you can see this is the cover of this great document on mentorship in STEM. And although it's relevant to STEM and particularly students in STEM, it is also relevant in almost any area of scholarship. So what is the definition of mentorship? We often talk about what is mentorship, and I think no matter what time we're in, whether it's this time or others, I think this definition of mentorship is the definition that was presented by the National Academy, and I think it's a great definition for any level. And it is mentorship is a professional working alliance in which individuals work together over time to support the personal and professional growth, development, and success of the relationship partners through the provision of career and psychosocial support. And I want to emphasize the word partners because we'll come back to this, that it's relevant to the development of the mentor as well as the mentee. So what does a mentor do? Well, based on all these resources that I've talked about, and particularly I'm discussing here maybe the more beginning mentor, a beginning scientist, somebody at an earlier stage of the career. So obviously the mentor has to play a role in educating the mentee in the current state of the research in the realm of scholarship, and provide resources to learn about the research topic or direction. And this, of course, in a time where some of our active research is not able to be ongoing, is a great time for mentees to become expert in a particular area. And I've also been impressed that when I send mentees off on the task of learning about an area, how much information they often come back with, information that I often may not be aware of. So this is a great time for the mentees to really get up to speed and be an expert in the field in which they're going to do some research with you. Of course, it's important for us to guide and discuss the ethical principles of effective mentorship and in research. And I know Dr. Perez going to talk some about this, but we have to play a role in letting them know what is really ethical research. And of course, outlining the research project itself. You would be surprised the number of times I talk with students and they're said, well, you know, I'm being told we're doing this or doing that, but I really don't understand exactly what the project's all about. And sometimes I think we may take it for granted that the mentee understands more than they actually do. So don't assume that they really understand all the aspects of the research project, where it fits into the greater literature, what we're actually doing, in this project and what's unique about it. So make sure that you spend some time outlining that. Mentorship agreement. This is in some ways similar to the data sharing agreement that was discussed earlier. And it's funny how many times I talk with people and they seem resistant to this idea of having a mentorship agreement. I think sometimes people feel that it's a legal document that will come back to haunt them in the future. But what is it? It's real important isn't to be a contract, and that's why we call it an agreement. But it's important to establish the expectations of the relationship. There's sometimes incorrect assumptions on either side as to what's going to be provided in this relationship. What will the mentor provide? I mean, we had a situation a while ago where the mentee felt that being mentored by the mentor meant that he or she could come into the mentor's office every day and discuss for an hour different problems. If that's what the mentor wants to provide and chose to do, that's fine. But if it isn't, it's important to establish just, what are the expectations of what is going to be provided. And then in turn, what will the mentee provide and be accountable for? What does the mentor expect of the mentee, and what are they willing to take on in the relationship? And again, as we were talking about, establish expectations for authorship of papers and roles on grants. And I would say, in an earlier era, in my era, we wouldn't have thought of, gee, there might be intellectual property involved in what we're doing, but that's a reality, as was pointed out. So what are the expectations for the work that the mentee does? Are they going to have mentorship? What is going to be their order in the author order? Are they going to be included in grant submissions? Again, I have had students in my class actually express some dismay that they felt that they should be part of an intellectual property application because they had done research in an area and they were not. So this can lead down the line to multiple difficulties. And as I think Cynthia mentioned, there's an article we share in my class, I think it was from Science, about the mentorship or the research prenuptial. That's why we need a mentorship prenuptial, if you will, as well. And of course, we have to make sure that the mentee has met all the regulatory requirements and standards. So have they done city training? It is possible to have undergraduates work with us in research, but unlike medical students, there are some requirements they need to undergo to understand HIPAA agreements and certification. So our role as a mentor is to make sure that the mentee has met all of these and also understands the importance of, why are we doing this? That this isn't just busy work, but we're actually learning important things about conducting research when we do this. Of course, as mentors, we have to model behavior. We have to exhibit in our own practices, as the mentee observes us, research best practices, ethical conduct of research. I like having my research students, whether they're medical students, fellows, residents, come into the room with me. My practice is generally to simply say, we have a research project. What you want to do, whether you want to be a part of this or not, is totally up to you. Doesn't affect our relationship, but I want to offer to you all the things we're doing here. If you're interested in it, we can have a research coordinator talk to you more about this now or later, and have the student observe that kind of interaction, and maybe then even stay on with the research coordinator to see just how the consent process takes place. I've actually had some students who became expert and trained well enough that they actually were able to very competently go through the process of obtaining consent with supervision, but doing a very good job. If you're still submitting your IRB, it's great to have them at least understand or be involved in the process, so they start getting on-the-job training of how does this work. And I've had some sophisticated research fellows who with guidance were able to do a great job of submitting an IRB all pretty much on their own. So of course you have to calibrate all of these things based on the stage of the student and their expertise. I've learned it's important that they really understand the process of placebo-controlled trials. I was the trainee in one case where I won't mention who the research mentor was, but I was meeting with one of the patients in a trial, and I mentioned, well, we don't know whether you're on placebo or active drug. And they said, what do you mean I might be on placebo? They were actually never told that they could possibly be on placebo. That's, pretty poor modeling of ethical behavior. So we have to make sure that the students are understanding that. And again, as I mentioned, teaching by example, allowing the mentee to observe and participate in the research project as appropriate and as they are capable. When we've looked and interviewed people for what are the characteristics of a mentor, the number one that seems to come up over and over is that they are honest and provide formative feedback. Honesty is always rated as the number one element of a mentor. So they have to be, you have to, provide feedback to the mentee. Are you doing this correctly? Are you not doing this correctly? Don't simply endorse or rubber stamp everything, but really understand, is the mentee doing things correctly? Do they understand what they are doing? And it's very important, probably the second thing to honesty is being open to their questions and their concerns. Not only is this an important educational issue, but it avoids understanding and misunderstanding and the risk of suspected research misconduct. There was an interesting article in the New Yorker a number of years ago about a postdoc who accused their PI of research misconduct. And the PI's view was this was the way things are usually done in this field of research, and that the postdoc didn't really understand that. Well, I think if the PI had been open to questions and the postdoc felt comfortable coming to the PI and asking about this, it would have absolved the PI of any suspicion of risk. We in my class talk a lot about an incident with Dr. Pody at Duke, who was doing incredible things in academic misconduct. And it seemed as though none of the students, fellows, residents, or other people went to the PI and expressed concern about what was going on, even though it seems that many of them did. And that means, there's a huge power differential between the PI or the mentor and the student. And that makes it even more important that we are extremely open to feedback and to understanding questions. And they may be pointing out as well something we're doing wrong. It's critical that if we take on a mentee, we commit the time to do this. I had a student just the past semester who asked me to give feedback on part of a manuscript they were writing. And their mentor said, I'm just too busy to look at this. You take a stab at it. Well, that's, of course, poor mentorship. So if you can't commit the time to really help your mentee do these things that they've never done before. Obviously, if they had a history of being an expert writer, that's another issue, but this was the student's very first research project. Amazingly, she actually did a really good job at writing it, but she shouldn't have been going solo. You have to be sure, obviously, that you have the resources to do the research project you're committing to the mentee. That may sound obvious, but I've seen cases where the mentor being eager to help a research student get something going, starting off on a project that like, oh yeah, I don't really have the funding to do that, or we don't have the resources to really get that done. So this takes a lot of forethought before, and is something else that I think has to be considered in the mentorship agreement. And of course, very importantly, supervising the conduct of the project, giving appropriate freedom though for experience. So I had, a phenomenal research mentor, Dr. Carl Lear. In my early research projects, we were pretty much side by side in doing the projects. By the time I was a senior fellow, I had designed a very complex patient-oriented research project and was very much doing a lot of it on my own, but Dr. Lear was always there. He came in to check in periodically to make sure things were going well. He was always available if I had problems. So calibrating your supervision with the skill of the mentee is critical, but they all need that kind of supervision. Obviously, guiding best practices in data collection, archival, and databasing. This is something we spend quite a bit of time in my class discussing, and really teaching the students that this is important. And this, again, is a time where a lot of active research may be down, but there may still be an opportunity for learning how to archive data and database. Teaching students that, gee, guess what? A lot of students think Excel is a database. It's not. It's a spreadsheet. Teaching them about things like REDCap and actual data collection best practices and good archival resources that they can use. And then guiding data analysis appropriate to the mentee's skill level. I learned a lot of my basic biostatistics by doing research projects and having Dr. Lear explain to me what is the appropriate statistical test and how to do it. And again, this is a great time to have students learn more about this. I later learned that it's even better to go ahead and formally study this, as in the MPH that I received or other training programs. But it's also important to guide the students. And again, sometimes students will really surprise you. I had an amazing student in my MPH program who we had to do some more sophisticated analysis using mixed linear models. And I gave her some resources and said, why don't you go read about this? Then we'll talk about how to do the analysis. And in fact, she came back a week later having read all the information and did the analysis completely on her own perfectly. So some students will pick up on these things amazingly quickly. Others, we may be at the stage where explaining, how a t-test works. But I think this is part of our role as well, to introduce the students to appropriate not only databasing, but how are we going to analyze the data? And then once it's analyzed, discussing the interpretation and meaning of the results. And to me, this is one of the really fun things. And again, something that during some downtime, we can spend time doing, discussing with the student, where does this fit into our current understanding of the field? What are we adding to the literature? Or what does this data really mean? Have some really good discussions with students, and they'll bring up their own interpretations that are often very insightful and sophisticated. So we have to commit the time to doing that as well. Being a writing mentor, this can be one of the most difficult tasks based on the mentee's skill and experience. Yet it's, I think, critical that they get the experience writing. And again, just jumping down to the last line as we discussed earlier, being sure in our mentorship agreement, we understand what role will the mentee have in the writing process. Are you going to be expected to write part of it? Dr. Lear's rule was if you did a majority or a large part of the work on a project, you could be the first author if you actually were able to write it and at least begin the writing process. And being a good writing mentor, which isn't always easy. My advice always is that it's important to be direct and specific. This is their first effort in scientific writing. And so Dr. Lear, again, was fantastic at that. I always thought I was a pretty good writer. An undergraduate, I always got A's for my writing, and this is really well-written paper. But obviously writing for the humanities or an undergraduate is very different than the field of scientific writing. So the first paper I wrote came back with lots of marks. Dr. Lear liked to write in a broad fountain pen ink with some subtle hints like, this paragraph sucks, or this sentence sounds like it was written by somebody who went to Ohio Wesleyan, where I went to undergraduate. But it taught me very rapidly what was good scientific writing and what wasn't. And it's very similar. I think one of the best books that guides us is a book written by Stephen King called On Writing, where he shares a lot of his experience with writing. And he mentioned that his best writing mentor experience was a 10-minute experience. He had started working in high school doing sports reporting for the local paper, and he took his first article proudly to the copy editor, who looked at it and just started making marks all over the paper, scratching things out, writing things. And he said in 10 minutes, he really learned more about writing than he did most of his life. So again, this takes time, but I think as a good mentor, we really need to do that. What does a mentor not do? Well, obviously avoid some of the bad practices of mentorship I've mentioned, setting a bad example, and avoiding what some people have called predatory mentorship, using the mentee as an expedient to get one's own work done without developing a mentor, or the mentee, I'm sorry, I should have said mentee. That happens more often than I think we'd like to. Sometimes it's overt, sometimes it's unintentional because we're not committing the time that we need to, but critical we don't lapse into that. What does the mentee do? Well, obviously following the mentor's advice and direction, following the expected timeline, respecting the mentor's time. Be honest as well. And when we do focus groups, honesty is also the highest ranked attribute of the mentee in everybody's opinion. Being honest if there are challenges to completing their work. So again, that's where the the mentor needs to be open and engages in the research relationship, contributing equally. Mentorship is not a spectator sport. So if we have mentees who are just sort of feel like we're taking their hand and guiding them along, I think we have to be concerned about whether we can really convince or continue this mentorship relationship. And this falls into an important category called reverse mentorship, where the mentee can teach the mentor. This was something that was really a concept that was started years ago by Don Welch, who was head of GE, where he encouraged his junior executives to mentor their older mentors about a variety of topics that they may be more expert in. I've had that happen to myself, and sometimes it's embarrassing. I had a really good MPH student doing a research project with me, and she'd been taking classes on good research practice and said, well, gee, Dr. Binkley, where's your data dictionary? You're always supposed to have a data dictionary. And I said, gee, I don't have one. So I learned that I need to do those things. And in fact, studies have shown that the mentor benefits from the relationship. The mentee brings new ideas and energy to the mentor. And there have been actually studies shown, at least in the business sector, that in good mentoring relationship, the mentor's career and career satisfaction grows as well as the mentees. So even mentoring new developing scientists and students, we gain a great deal of benefit. And we continue to grow as mentorship. And mentorship training is never ending. The CCTS training program in mentorship is available on the FD4Me website maintained by FAME. We have 3 modules for mentorship training. Many of you are probably aware of the National Research Network, and this is their website. Noah Weissletter in the postdoc office is a really good source on when they're doing training and when that's available. And we grow from each mentoring relationship. What are the barriers? Well, do you have the time? Do you have the resources? Do you have the support from your division and department to do that? Are you ready to be a mentor? Most people are probably more ready than they think. But again, this is where the training is very useful. I just want to mention a resource if you're mentoring junior faculty and also to involve yourself in mentorship. This is a program called FAME Pro, which FAME does in collaboration with the CCTS. It invites new faculty investigators to submit a research idea. Those selected can use their current research mentor, or we help them in identifying a mentor, and we're always looking for mentors. They receive training in biostats, study design, and other training, and the graduates have been extremely successful in publications, funding. Many of them have, some of them at least have gone on to receive Davis-Brenner grants. How do you know you're succeeding as a mentor? That's an area of active research, and there are many tools available in the Academy report, but I think the success of the mentee is probably agreed to be maybe the best metric. What's different in the area of COVID? Well, in many ways, not many things. All the expectations we have are often listed, exist obviously in the current time. The principles of the mentorship are the same. We may have some more time at the present time to devote to some of the things that I've mentioned. But there are new wrinkles. We have new risks to explain to the patient about how are we going to protect them from COVID exposure? How do we minimize these risks? How do we recruit patients to research programs in a safe environment? And these are all things that we can now teach our research mentees in a new era and make this truly a learning experience. So I think I'm right at 10:25 and I'm at the end of my slides. So thanks everyone for listening. And if we have some time for questions, we can take those.
Please offer up any questions you might have. Abby mentioned the interesting situation now in this crisis, maybe not being able to carry on some of the work that you're talking about as a junior faculty and getting behind in some of those commitments. What kind of advice or how would you mentor someone in that situation?
Well, that's a great question. And actually, a couple of weeks ago, I did a power hour for FAME about scholarship in the time of mentorship. I won't deny that it's a challenge, but it's also an opportunity to include trainees in this entire process. As I mentioned, even though I have administrative and clinical responsibilities, can I include the mentee in discussions about how this is going, how we're trying to resolve this? Learning about, this is a time to learn about how do we you know, engage in a time that we've never seen before. And this will probably not be the only time in the mentee's career that they encounter something that's never been encountered in their training. So trying to take the time to involve them in what you're dealing with, and that can help them prepare for whatever future things that are so different that they may encounter in their future careers. Any other questions or advice from some of the, I know there's some experienced mentors out there.
Phil, I have a question. This is Abby. So I really wholeheartedly agree with your comment that if you're a mentor and you have taken on a trainee, you owe them your time, you owe them your energy and commitment to seeing them through this process. But I do think COVID-19 has thrust a lot of mentors into new leadership roles where like something's got to give. They just don't have the hours in the day to do the things that they committed to doing. I mean, it's not just mentors. This is true for all of us. So how should mentors and trainees kind of manage those tensions? There are lots of learning opportunities, but there are also some things that just have got to be shut down for this kind of crisis period for some people.
Yeah, well, I think part of that is, first of all, if you're thinking about entering a new mentor agreement, and this is just like any time you enter a mentor agreement, are you going to have the time? So you may have to tell someone, you know, I really can't devote the time right now. And I think that's one of the most responsible but difficult things that a mentor can do under any conditions, whether it's now or otherwise. It's very flattering, obviously, to have somebody ask you to be a mentor. It's a real sign of respect. But so you have to have the honesty as to whether or not you can do it. If it's an ongoing relationship, you may need to recontract a little bit. Like, so my time's really, so can you think of some things that the mentee may be able to do without your direct supervision? Some of the things we talked about. Can they get further expertise at your guidance in biostats, or archiving data, or interpreting the data, or have them start to write up a piece of data just to start practicing writing? So I think we have to think of things that we can have them do a little more independently that can still really grow their understanding and career, that may not so intensely involve us. That's a great question. I'm not trying to minimize the challenge. It's a real challenge, but those are some of my thoughts as to the approach. Stuart, I think you're muted.
I am muted. Thank you. Thank you very much, Phil. And at this time, we have scheduled a brief break or intermission in the program. So can refill your coffee cups and get some refreshments, which we would provide if this has been an in-person presentation, but you'll just have to take care of that on your own. And we'll be coming back at 10:40 with a presentation on some of the ethics, the whole segment next really on kind of the ethics of research at this time. So we'll take a break and then we'll come right back. So please stay with us. As we are coming back together again, and starting a section related to the ethics of research, I wanted to point out that the OSU Center for Ethics and Human Values, as part of their CARE program, which is Conversations about Research Ethics, is presenting this program on ethics and regulation of clinical trials for COVID-19 treatments. It's going to be on May 20th at noon. And you can see who some of the presenters are here, with professors of law, nursing, and ethics and philosophy. I'll be sending out a link, this brochure and a link with further information for you after this program, but I wanted to call this to your attention. Our next presentation is a pair of speakers talking about beneficent human and animal research, and we'll begin with the theme of beneficent human research with Michael Pera, who is Professor Emeritus in the Division of Infectious Diseases in the College of Medicine, with extensive experience running AIDS clinical trials. And he's the director of the Center for Clinical and Translational Sciences regulatory programs, as well as their participant research and recruitment programs. Michael is currently working with doing rounds in the hospital. He will join us for questions, but has pre-recorded his program, and so I will turn that on for you now. We seem to be having some technical difficulties. Please stand by. Oh dear. Still on.
Thank you for attending today. My name is Mike Para, and I'll be talking about beneficent human research, or as I decided to title this, Clinical Trial Tribulations During the Pandemic. This is a perfect storm. It's a new infectious agent, transmitted and acquired just by breathing. There was nosocomial transmission with a desperate need for PPE. There was exponential spread where one person could infect 2 or 3 others, and then they could infect 4 or 8. They could infect 16. There was significant morbidity. The combination of those two of explosive spread and morbidity brought heavy demands on our healthcare resources, and there was societal disruption. This was a new infectious agent. There were no effective therapeutics, but because tissue culture is not very useful, there are many wannabes that people were suggesting as effective therapies or potentially effective therapies. The pandemic brought a do something now attitude. As you know, the pandemic started in China. It was an unknown disease, unknown pathophysiology, unclear spread. They were suddenly overwhelmed by the medical care required. Unfortunately, this led to some poor quality science in the midst of the crisis. That led to poor quality publications, rapid manuscripts review, if there was any review. Manuscripts were usually released in electronic form, often labeled peer review pending. The lay media picked these up and reported results before the manuscripts were even reviewed. There was a do something now attitude. That was true with respect to clinical trials. As the disease subsided in China, clinical reports flooded out, reports having just 2 or 3 patients flourished. A lead article in JAMA reported on 5 patients. An article in the PNAS reported on 10 patients. In these studies, there were often multiple agents being used simultaneously, and what to attribute harm or benefit was unclear. Social media and the cable news rapidly spread questionable results. And then, of course, we have the political commentary with little scientific input, which made us all crazy. What I'd like to do today is discuss 3 therapy trials showing the good, the bad, and the ugly of trials during the pandemic. The first trial is the ugly. You look at this plot and you say, I think I've seen that before. Well, I'm sure you have on the news. This was the first plot related to the use of hydroxychloroquine and azithromycin for the treatment of COVID-19. It was an open label, non-randomized clinical trial, and you look at that and you go, wow, the placebo or the control group, very little resolution of the infection versus the azithromycin, which was basically undetectable in 5 days. You see the p-value up there, and you might be tempted to tweet, if you tweeted, this is a game changer. Unfortunately, when you start looking at the study, you find something else entirely. While hydroxychloroquine did have activity in vitro, whether it worked in vivo was entirely different. There were some reports from China, but actually nothing in print, that reportedly it was effective. In this study, the treated subjects had COVID-19 with a spectrum of illness from asymptomatic to quite sick, and they were from Marseille. Interestingly, the controls were either people who were ineligible for the study or from 4 other cities being cared for by other clinicians. The protocol included daily nasopharyngeal swabs for PCR among both the treated and controls, and the endpoint was the PCR positive for virus on day 6. They had sample size calculations, and there were supposed to be 24 patients in each arm, and this is out of their article, we enrolled 36 out of 42 patients meeting inclusion. And since there were 26 treated, that means there were 16 controls, of which 10 were eligible for the study, would have been eligible for the study. The results, at baseline, the clinical status of both the treated and controls were similar, but strangely, 6 people that were in the treated arms exited the study early. 3 got sick enough to go to the ICU, 1 died, 1 had drug toxicity, and 1 left the study. All these 6 were excluded from the final analysis, which is very strange. I mean, you could extrapolate that by saying if 19 of the 20 treated died, but 1 survived and was undetectable, that this drug was 100% successful, which makes no sense. Or as I like my never mind, I won't give the football analogy. So 20 were treated were in the analysis, 6 on hydroxychloroquine, 14 on the hydroxychloroquine, 6 on azithromycin. And the results on that plot, 14 of the 20 treated were undetectable at day 6, but only 2 of 16 of the controls were undetectable at day 6. As I said, the analysis excluded 6 treated patients who dropped out before day 6 because of progressive disease. Also strange was among the untreated, 5 were counted as infected, although they lacked the day 6 endpoint PCR. There were a couple of other things with the manuscript about a very suspect PCR assay that did not look accurate. And then the authors reported a confirmatory study which had inconsistent results from the first study. The good news here, though, is that there are over 25 placebo-controlled trials of hydroxychloroquine and azithromycin for COVID listed on clinicaltrials.gov. There's a huge placebo-controlled trial for the prevention of COVID in 3,000 healthcare workers and first responders. And Dr. Fauci asked the AIDS clinical trial group to quickly develop a protocol and implement 2 large 1,000+ patient placebo-controlled trials in ambulatory and hospitalized COVID patients. So some good will come out of this. The next I'd like to talk about is convalescent plasma for COVID. I regard this as the bad. Straightforward rationale. Plasma from recovered COVID patients should have viral antibodies, and infusion of this should neutralize virus and lead to improvement. And there are some historic studies, even going back to the 1918 flu, that serotherapy is beneficial. And there are some uncontrolled trials for MERS and SARS that showed some help as well. So in this attitude of must do something, the Chinese did something. They published a paper. As I said, it was a lead article in JAMA, published online. 5 COVID patients on ventilators received 2 units of convalescent plasma. As you'll see, on the next slide, these patients, here's the 5, all received methylprednisolone, all received some form of an antiviral that may or may not have activity, but was included in the report. The other thing that's notable here is that all patients had symptoms for at least 3 weeks before they came into the hospital. So what were the results of this study? Well, the results published in JAMA were that after 2 weeks after the infusion, 3 left the hospital and 2 were improved on the ventilator. Who can tell if plasma did anything here? I mean, there's nothing we can compare it to. The only thing this did was give the authors a publication in JAMA. Also interesting was there was an editorial in the same issue which stated if the results of a rigorously conducted investigation, such as a large-scale randomized clinical trial, demonstrated efficacy and included data supporting biomarkers of success, this therapy could help change the course of this pandemic. Such biomarkers would accelerate our vaccine efforts. So where did this take us? Unfortunately, 3 days before this data, and that is any data on this was published, the FDA approved an investigational convalescent plasma for patients if their O2 sat was less than 93%. And their rationale was, since no known cure exists and a vaccine is more than a year away. Any licensed physician could request it on an emergency IND, and the plasma could be provided by any blood bank that had it. Within a week, the FDA arranged with the Mayo Clinic to establish an expanded access protocol. Patients in an acute care facility meeting the criteria above could be given plasma. All that had to happen was the patient needed to sign a consent that this was an investigational product. Minimal data was collected on these patients, just days of hospitalization, days on a ventilator, days in the ICU, and death. There was no control group, but the data, but in this group, they also collected the data on those who consented, but in the end did not get infused with the therapy. So what has the Mayo Clinic Expanded Access done? As of today, 2,200 sites are approved, 5,700 physicians are approved to order the plasma, 15,000 patients have been prescribed it, 10,000 plasma units have been infused, and in Ohio State, we've given over 30 units already in the last 7 days. And still, we have no idea, after all this, if this does any harm or good. And I don't know that we're going to have it. The not quite so bad is about 60 trials are listed on clinicaltrials.gov using convalescent plasma. Sadly, only 10 of these actually are controlled trials, and only 3 are controlled using non-convalescent plasma. Will this FDA-promoted early access protocol subvert the controlled trials so we never learn if this works? That is a potential. All right, finally, remdesivir, trial 3, the good. Do something helpful now. Remdesivir is a nucleotide analog which inhibits RNA polymerase, and it's a daily parenteral agent. It's made by Gilead, who developed some blockbuster nucleotides against HIV and HCV. It's worked in primate models, and it's appeared safe in human studies when it was tested for Ebola. Gilead initiated 2 placebo-controlled trials in early February in China, large studies of severe and moderate disease, but sadly, they didn't have enough patients by that time to finish the trials, and so they ended with less than 50% accrual and gave inconclusive results. I think what's most amazing is the NIAID initiated an adaptive COVID treatment trial, ACT, on February 23rd using remdesivir. At that time, there were only 15 cases of COVID and no deaths from COVID in the United States. They knew what was coming. The trial was an 800-patient placebo-controlled trial for people with an O2 sat less than 94%. Gilead also, about a week later, initiated 2 trials, but they were basically a remdesivir. Everybody got remdesivir, but it was 5 days of treatment versus 10 days of treatment in people who had severe but not on a ventilator or moderate disease. And then for people on a ventilator, Gilead opened a limited compassionate use protocol that was reported in the New England Journal April 9th before any of the results of these other things. And the number of authors exceeded the number of eligible patients, which was 55, and 68% of the patients improved. But the study's bottom line was, we need a controlled trial. On March the 27th, remdesivir compassionate use for ventilated patients was made available in the United States, and we're using that now. Now, about 2 weeks ago, the NIAID released the DSMB report, which said the median time to recovery from severe COVID, 11 days in remdesivir, 15 days in standard of care. That was a success. Remdesivir helped. Survival looked better as well, but it was borderline. Nothing else has yet been reported. Simultaneously, the SIMPLE trial from Gilead showed that 5 days of treatment was as good as 10 days of treatment, and those treated early did better than those treated later. So on May 1st, the FDA authorized emergency use of remdesivir for COVID patients with O2 sats less than 94%. And the bottom line, even during a pandemic, good science wins out. Thank you.
Thank you, Dr. Para. Our next speaker is Peter Reiser, who is the chair of the Institutional Animal Care and Use Committee at OSU, as well as the professor and chair of biosciences in the College of Dentistry. Dr. Reiser. Dr. Reiser, are you there? Please stand by as we work our way through the perils of Zoom technology. Well, I'm not sure what has happened to Dr. Reiser. Again, please stand by. I think at this point, we will turn the program over to Alex Sparaboom to address issues in conducting ethical research. Dr. Sparaboom is a professor in the Division of Pharmaceutics and Pharmacology in the College of Pharmacy and has served on university committees investigating academic misconduct.
All right.
Is the audio okay?
Yes, it is.
Okay, great. Well, thank you, Stuart, and also I'd like to thank Cynthia for inviting me to give this talk today. So I think some of this material that I'm going to share with you has already been captured in some of the previous talks, but it may serve as a reminder, if anything. I was a little bit worried doing this, because of computer problems that we've seen already with Cynthia. And I was reminded of this also when I opened up my SCI reports that came in yesterday on a course that I did last month that was completely virtual, where the first comment from a student was that Dr. Sperboom is not very good with computers. So I got a bit worried doing this today, but we'll see how it goes. So these are just the typical disclaimers I put in, and I think only one is relevant, which is this one, that whatever I'm going to say today is not necessarily consistent with official views of the institution. So with that, let's go and get started. So the question that I was asked to address specifically is on this slide, and it is how we can, as OSU investigators, guard against collaborators either locally or elsewhere who are committing fraud. How can we protect ourselves if this happens, but it won't be uncovered until next year? And the short answer to this question, at least from my perspective, as an investigator at OSU, is that we just maintain the typical strategies that we think are already in place. And so we having thought about this in a bit more detail, I think many of the things that we already have in place are just in our opinion sufficient to ensure sufficient protection. And so I'll go over some of these things towards the end, but maybe just for clarification, outline some concerns that have been raised globally by a number of research integrity offices worldwide, including in Europe, where there's been voiced serious concerns, and that particularly in relation to this COVID-19 business, that this may be more important now than ever before. But nonetheless, there's just the typical urgency to the research community to respect the common integrity standards as they have been in place all along. Specifically related to, of course, the COVID-19 outbreak, has this, business about a perpetuating perception that when it comes to, rigors of science, that, there's some situations that demand exceptions. And we've just heard about some of these in relation to clinical research that really affect standards of quality. As to whether or not, this is also relevant to issues beyond COVID-19 related research, I'm not expressly familiar with, but we can talk about this in a little bit more detail. So with regards to specific things that we're talking about, I thought it would be useful to include a few definitions that are commonly used for some of the terminology. So research misconduct in this context will then be defined as fabrication, falsification, or plagiarism in proposing, performing, or reviewing research or in reporting results. And so as part of this definition, fabrication is typically referred to as a description of experiments that are not actually performed, or invention of data not actually collected. And then falsification most commonly will then be referred to as manipulating research results or equipment or processes, or changing or omitting data or results such that the research is not accurately represented in the research record. So a number of related terms that are associated with falsification are commonly referred to as cooking and trimming, given the definitions are given here on the slide. And if you look at this in terms of, going from likely mostly occurring to less frequently occurring, I think this goes, from top to bottom, with fabrication likely to be the least, common towards the most common plagiarism, which is typically defined as reusing someone else's words, ideas, or results. So the OSU policy and procedures concerning research misconduct also have their own definition that you can find on the webpage, shown on this slide, defined as the appropriation of ideas, processes, results, or words of another person without giving appropriate credit. Now, the good thing about this plagiarism business is that, at least with regards to the reuse of words, this can be, very efficiently and quickly tested for using specific tools that are now available free to everybody, including software such as Authenticate, that can be utilized by everybody employed at OSU, including students. And the link to this software is actually given, if you follow the web page that is given here. Now, if you prepare documents for publication, it's very likely that also journals will utilize similar software in order to identify the reuse of, previously deposited, phrases. So here's an example of, what documents may look like if you run them against existing documents with color coding indicating, specific reuse of phrases and sometimes entire paragraphs. So if you submit a paper and it is being returned by an editorial office looking like this, then that really means you have to, change it to a very substantial extent in order to, avoid, having this accusation of plagiarism. I think the current standard is that, less than 20% redundancy in text should be observed when submitting new works for publication. So in addition to the reuse of words, I actually was reminded preparing this slide set that I had observed a long time ago, at least not so much a textual duplication of some of our own publications, but at least conceptually a story that was virtually identical to an old paper we had published, which was in itself not very noticeable and interesting. This was related to a story we did, what is it now, almost 23 years ago, on the influence of a formulation excipient in what at the time was the hottest drug in oncology, Taxol or paclitaxel, and its influence on the ability of other medications to be toxic to bone marrow cells. And so this hardly interesting story was basically, at least conceptually, almost entirely duplicated a few years later by a group in Egypt. And so even though running these two particular papers on Authenticate indicates, a mere 9% direct duplication of text, this was in essence, a virtual copy of, the story we had done. I did bring this to the attention of my department chair at the time, but I was told to back off because these people in Egypt apparently had a lot of trouble with their funding, and I was just told to leave this aside because these people were having trouble getting their research going. So much for that. Things have changed in the meantime, I think. At the same time, I would like to point out that this whole plagiarism business, especially with respect to the reuse of text, has also triggered a lot of discussion. Much of this online from people that have differing viewpoints on the whole thing, and so there's a very entertaining page you can find if you plug in this title in a Google search and then find this piece by Stephen Dutch, an investigator at the University of Washington, who's of a very different opinion about the seriousness with which we should take this whole plagiarism approach to the publication of scientific as well as non-scientific literature. And so he thinks this is all a bunch of bogus, and he gives really good examples of where people like famous authors like Ian McEwan are being raked over the coal for not using quotation marks, certain sentences in their books like Atonement, and then get attacked very heavily for committing plagiarism when this may be just a bit overstated. And I think I would like to give one other example, which is, in the context of writing review articles, I have always been of the opinion that if it's the intention in a review article to accurately and faithfully communicate, prior ideas, whether they are from ourselves or from other authors, then what's better than to use the exact words that were used previously. And so this is obviously now a big deal because even in a review article, we're not allowed to go reuse prior statements from authors, at least not to any substantial extent when it considers the total of the words used in a paper. The other thing I'd like to point out, which is kind of interesting, and I'm going off on a tangent now, I realize, but it's I thought this was very interesting, which is the term plagiarism originated, as you may know, from ancient culture, specifically in Rome. And they didn't have a big deal with creative imitation to the same extent as we do now, or at least as do many scientific journals. And in fact, it was in many ways it was encouraged, as long as it was not considered a symptom of laziness. And certainly it was not denounced simply on the grounds of being unoriginal. And I'd like to point this out because if we think about this in a broader context, this originality business in the literatures is something that's really relatively recent. And I thought long about giving a good example of this. And I think that this is something very dear to me, which is, as you may know, verbal agreement between the Synoptic Gospels in the New Testament. There's some, this has been known for hundreds of years, of course. And here's an example from, Mark Goodacre's book on the Synoptic Gospels, where, he points out that there is long stretches of text that are just verbatim copies between the different Gospels. And we don't really know why this is. And, of course, as you may know, people have, hypothesized every possible idea as to how this could be. But my point is not so much to, to advance a particular theory about this idea, but more so that if you were to plug in the text, the Greek text of the, New Testament Gospels into a software package like Authenticate, then, there would be a stunning degree of, verbal similarity, to the point that, if this had been a strategy in the past, let's say in the 1st century, then, 2 of our 4 Gospels would not have been preserved. And I think that's just a completely absurd idea, right? So this is just as a piece of context, that we need to really consider also in viewing this as something of relevance to the scientific literature. And the same thing is true in music. In music, at least in older music, there was not really a big deal to plagiarize. There's tons of examples where, people could just take, a new text to an old tune and then, consider it a new composition. And we also have to think about things like, again, something really dear to me, that many of our great works of art, in music at least, were derived from existing material and, applying currently adopted definitions of plagiarism, we would be devoid of some of the great masterpieces in music, right? So something to stew on, I think. Now, back to science now. One thing that led me to think about this in a bit more detail, researching this, was this idea, how prevalent is this really? And is this something that we need to be concerned about when we don't have access to our labs? And so this somewhat surprised me. You may be familiar with it, I was not. One of which is that many people in the field, in the biomedical field, have actually firsthand knowledge of misconduct to some extent. And, in many scientific research articles where misconduct surveys were conducted, it turns out that up to 2%, which is a pretty substantial number of scientists, are admitting to having themselves fabricated or falsified results at least once. And then consistent with my own experience, even though this has been very often reported informally, to supervisors, it's very uncommon, that this type of behavior is actually leading to formal complaints. Again, something I think we need to worry about. So why do we care about this? Well, we care about this for a variety of reasons. One of which is, of course, that this was really the foundation, ultimately, this honesty, for leading to the development of the scientific publication world as a scholarly enterprise, leading to, the idea that, competition is important, being the first is important because it's a measure of prestige. And, this led to ultimately help other scientists to develop new hypotheses that could, be built on those prior discoveries. And most importantly, because I think, ultimately science is, of course, a fundamentally, cumulative enterprise where, people build upon prior work. And for that reason, of course, we all have a moral obligation to disclose new discoveries that are trustworthy. And then ultimately, this is, of course, essential because that dictates in the end prospects for people, and as well as prospects in relation to promotion and tenure. The thing about, the scientific literature as it's currently being perceived is, that independently of the idea of there being issues with falsification, there's, an issue associated with reproducibility. It's not entirely clear why this is, but I'm sure all of you are very well aware of these stories that came out in Nature a few years ago, where, there's been an alarmingly high percentage of scientific discoveries that cannot be reproduced or replicated by other investigators. And this is as high as 90% in a field such as chemistry, but up to 70 to 80% in the biomedical field. There's many reasons for this, but some people, some commentators have argued that this really has led to science taking a turn towards darkness. And so what can we really do about this, and what are really the causes for this? This is not a particularly interesting slide, but I put it up because it's an example of a plagiarized slide. I didn't make it. But I think what we can learn from this is that, many of the reasons that people have speculated as to the cause for this are related to something that, Phil talked about earlier, which has to do with best practices in education and mentoring. So what can we do about it? This is, a couple of generic ideas that people have come up with recently, one of which is related to, making data available. There's now many journals, as you know, that require raw data to become available. Hold journalists accountable. This is something that was just mentioned in the, trials that have been reported recently, on the COVID-19 related research. And then very less likely, but an interesting idea that I hadn't heard before is the possibility of, in the future, pursuing anonymous publication, so that that can be really a focus on new findings rather than, on curation of a career. So yeah, holding journalists accountable is, of course, an important issue. Likewise, this has already been addressed again in one of the last slides. This is now more explicit, with names attached to it. This is not to ridicule, anybody in particular. I'm sure and positive it would have been, the same story had it been another administration, but this is a very serious problem, of course. So what can we do about it? So this is, some of the thoughts, that we have gone through. Most of this has to do, again, with education, insisting on preparing well for things, insisting on reproducibility, collaboration emphasis rather than competition. This is something that we've started to do a little while ago, is the solicitation of pre-publication peer review. This ultimately, of course, always leads to delays in publication, and sometimes by many years, but has been, for us at least, very rewarding in terms of improving on the work that we are intending to submit for publication. And then again, something else that was brought up again by Phil previously is the idea that maybe we need to reconsider approaches that we take to our strategies with respect to data analysis, statistics, and data presentation. So we got to really think about this, quite extensively, after, we were involved indirectly in terms of, being a member of an investigations committee in a high-profile case at the College of Pharmacy. And of course, this current COVID-19 related closure has, given us an opportunity to really think about, what can we improve and how can we make things better. I maintain, of course, that, a key answer lies in, further education and, communicating the basic principles of science is really key, to making things better. So what are we doing particularly? We continually educate employees and students on good practice, ensuring that there's proper record maintenance. We'd like to think we try to improve on training, promote mentorship, as discussed by Phil previously, along similar lines. We increase interactions. In the pre-COVID-19 days, I used to go into the lab on a daily basis, have discussions with people every day. That's no longer really possible, but we try to keep in touch as much as we can, to keep discussions going and, do as many presentations as we can. Now, this repeatability thing is, of course, essential to us, and we've implemented a number of strategies to address that question. One of which is to, insist on validating observations in secondary models. I'm not going to go into great detail about this. This is just one example, where we went through the trouble of replicating an observation made in a particular animal model, in mice to be exact, reproducing this in rats. And so this is a genetically engineered model that we had to develop, and that caused tremendous delays in us pursuing this. And this example is not given here for you to try to absorb the numbers, but rather to appreciate the idea that replicating observations in multiple model organisms might be very useful in order to be convinced of some worthiness of the data. Another thing that we've implemented a while ago is solicitation of external validation, especially when it comes to things that are unexpected coming out of a lab observation. Here's an example of which we were interested to reproduce an observation in which we found that the ability of cisplatin to cause high-frequency hearing loss was dependent on a particular transporter being present or absent. And in order to to verify that this was a real observation, what we did is we shipped these animals to another lab in Germany and asked them if they were able to reproduce it, and they were. In fact, their data looked better than ours. I'm going to skip this. There's several examples of this where we went through the trouble of shipping reagents to sometimes even our competitors with the request for duplication of data in order to guarantee that we were on the right track with something. And I think this is something that we will continue to pursue, even if it will cost us a lot of time and ultimately will delay further publication of data. What we are currently doing in terms of data analysis, this is of course a great opportunity to go through detailed data analysis. What we're doing is we have existing data analyzed by at least 2 people, and then one of these people will not even know about the project. And I think this is a strategy that introduces an opportunity to take out some bias in the resulting information that can be generated from projects. In the end, the responsibility lies with PIs, and so for me that means that ultimately prior to publication, I cross-check and double-check data and recreate all figures that go into publications in order to ensure that they come out the same as those that are being inserted into a draft manuscript. So again, this takes a lot of time, but it is worth doing in order to guarantee accuracy of information. And again, this is something that Phil brought up. Can we use this time to learn some new things that we didn't know yet in terms of how to properly analyze data and present data? And so, for us, what we've learned here is that, this business about staying home may, of course, induce researchers to, ignore due consideration for ethical standards. This applies to all research, at least theoretically. We know this is certainly true for COVID-19 related research. I think, the rules that have previously been established by the scientific community, to ensure robustness and trustworthiness, we need to continue to follow those even under difficult situations, because otherwise, this will distort progress. Ultimately, PIs are, ultimately responsible for this, and they need to be particularly vigilant in identifying, potentially fraudulent practices. I think the strategies that we have in place are relatively easy and straightforward. If those are being applied, stringently, I just don't see how there can be any, fraudulent processes, coming out of the lab. That seems to be, virtually impossible, but not something that, may be of interest to other labs to implement, considering, the time it will take to take away from, making progress particularly. And I think, ultimately we all agree this is important, because if we don't take this seriously, it will, ultimately influence the integrity of our own research. So if you want to know more about this and, get in touch with, the Office of Research, here's some, good information to go check out. I know Susan is on the phone as well on the call today. She may, be available to answer some questions as well. And so with that, I'll thank you for your attention.
Thank you, Alex. And we have resolved the technical issues with Dr. Reiser, and he is able to join us now. Are you there?
Yes. Can you hear me okay?
Mm-hmm. Yes.
All right. Okay, go ahead and proceed.
Proceed.
Okay. So thank you and Cynthia for providing an opportunity to provide information about the OSU Animal Care and Youth Program during the current pandemic. Several changes are in place during the state of emergency that ensure the beneficent use of animals. These changes are directed toward ensuring the welfare of animals on studies and, very importantly, ensuring the safety of individuals in our animal facilities on campus. These are two major responsibilities of the Animal Care and Youth Program at OSU at all times. And changes simply needed to be made during the pandemic to be able to fulfill these. The responsibilities are normally shared by many individuals. Those who use animals in research and teaching, the Institutional Animal Care and Youth Committee, or the IACUC, and University Laboratory Animal Resources, or ULAR, to maintain an accredited program. So I have a list of changes that were implemented that directly impacts maintaining the welfare of animals during the pandemic. The first is that Dr. Valerie Bergdahl, who is the director of ULAR and the OSU attending veterinarian, has managed the ULAR staff who provide daily care and monitoring of animals so that adequate levels have been maintained and continue to be maintained. Dr. Bergdahl recently indicated that no major problems in the animal care program have been encountered so far during the pandemic. Quite significant changes have been made in terms of the logistics of providing care and monitoring animals, but no major problems have been encountered. Continuing with some of the changes that impact animal care and monitoring, no new studies involving animals can be initiated. And this is to minimize the number of individuals performing animal-related activities and follow-up monitoring of animals. Importantly, no new animals should be enrolled into ongoing studies. I think this was a point of confusion for some individuals. They thought that because studies had been started prior to the shutdown, that they could continue to enroll new animals in ongoing studies. But clearly, people were expected to not enroll new animals in ongoing studies, as well as to not continue the ongoing studies unless they fit the criteria for critical research. Monitoring of animals can occur, but this must be approved at the college level and in coordination with ULR staff. No new animal orders are being approved unless the orders are for what is considered to be critical research, such as that related to the pandemic. The benefit of this is that it reduces the workload on ULR staff and allows the staff to focus on a smaller number of animals. So far, there has been a decrease in the number of animals of about 10% since the beginning of the shutdown. Related to this, ULR staff were available to provide monitoring of animals when individuals, study team members on protocols cannot do this, such as following post-surgical monitoring. If study team members cannot provide this, arrangements can be made with ULR so that ULR staff can provide the monitoring. Activities that are related to maintaining valuable animal lines are allowed, but these, again, must be approved at the college level. These include setting up breeding cages and tissue sampling for genotyping purposes. Maintaining existing cell lines is also allowed as indicated by through the guidance from the Office of Research, but this is not overseen by the IACUC. I just wanted to mention that maintaining existing cell lines is also allowed. There was a webinar that was recently offered, I believe it was last week, that focused primarily on understanding and following IACUC policies related to animal surgery. Now, this was prompted by results of semi-annual facility inspections and discussion with study team members regarding their protocols. It was not specifically related to the pandemic, but it does relate directly to today's webinar, which is focused on maintaining research integrity through the use of animals. It was offered at a time when many individuals were working remotely and hopefully had an opportunity to adjust their schedules to be able to participate in the webinar. This webinar was recorded and should be available to others for viewing. People can contact the IACUC office for information on that. Continuing with the changes that directly impact animal welfare, if the IACUC receives reports or questions about specific activities from ULR staff or anyone for that matter, the office staff and committee members act accordingly, following formalized office procedures that are always in place regardless of the conditions on campus. Fortunately, during the pandemic, there has been some but very limited activity related to this point. So those who are working with animals remain, for the most part, compliant with their approved activities. The Institutional Biosafety Committee has been meeting twice weekly to evaluate and approve pandemic-related activities. This is in addition to the IBC regularly scheduled meetings, monthly meetings. I mention this because IBC approval needs to be granted before IACUC protocols and amendments can be approved. Let's see, what else do I have here? Oh, if the IACUC receives reports or questions about specific activities from ULR staff, I already mentioned that, how the IACUC acts on those. We have compliance subcommittee that has been working on several instances that have arisen during the pandemic, but that aspect of the committee function is also being maintained. Finally, the semi-annual facility inspections that are conducted by the IACUC are on hold for now, and plans to resume those inspections once we are allowed to return to campus are being made now. So what are some of the changes that have been made to ensure the safety of individuals working in our facilities? Well, the ULR staff were initially working on split shifts to promote physical distancing and now are working on staggered, partially overlapping shifts. So there is less contact between individuals at the start and end of shifts by having a staggered work schedule. As a consequence of the guidelines provided by the Office of Research regarding what is critical research and allowed to continue, and the guidelines provided by ULR, there has been a decrease of about 90% in the number of individuals entering the vivarium on campus. Therefore, many people are following the guidelines, and this has promoted physical distancing. This allows ULR staff to focus on additional monitoring of animals that they are doing when study team members cannot do this. Individuals who are approved to work in vivaria are always asked to coordinate their time in the facilities with ULR staff to ensure that adequate supplies are available at all times and to promote social distancing. So overall, the assurance of animal welfare and the safety of individuals working with and caring for animals has been maintained with careful considerations and the implemented changes during the pandemic without any major issues. Thank you for your time and attention.
Thank you, Peter. And here's a question for you. Is the IACUC still reviewing, approving protocol amendments for non-COVID research?
Absolutely, yes. Actually, I was going to try to fit that into my list, but that's not a change. We have been very fortunate as a committee because a lot of what we do is done remotely normally with the e-protocol system that the IRB shares as well. And that has all been going on. We haven't had any problem finding reviewers among the committee members who have time to do this. So I sense, but I don't actually have hard numbers, that the number of items that are submitted, protocols and amendments, has decreased modestly. That's just my impression. But all that activity is ongoing.
There's also a question that some vivariums do not have face masks, but are requiring them. How can you address how shortages of PPE are being addressed or planned for?
So I know they have, right now I heard that they have adequate PPE for the vivaria. Perhaps the distribution to the different vivaria needs to be addressed. But if any individual who is out there has a concern, they should contact ULR directly and ask them about this. As far as I know, adequate supplies are available. The cost is escalating, as you might expect, and that could impact ULR-related charges in the future. But right now, there should be adequate supplies.
Also, Valerie Bergdahl mentioned at a program a few days ago that people going through the hospital have to be checked, and they issue the mask, so you could take that mask with you to the vivarium.
Yes, I know. So the vivarium in our building, Postal Hall, we're asking people to enter through a hospital point of entry so that they can be screened and given a mask.
Other questions from our participants? For Alex, Peter, or Mike? If not, I want to thank you all for attending, and especially thank all our presenters for their very helpful information. I hope that those of you in the audience did find it helpful. I will be sending out some of the resources that were mentioned here. If you don't get something today, send me an email. I may have missed your email address or didn't register in time. So if you missed some resources, just let me know and I will make sure that you get them. Also, look for a short evaluation survey to come to you tomorrow morning, and we appreciate your feedback. It helps us to plan programs like this and to provide information that you need to carry on your research. So again, thanks everyone for attending. Thank you, presenters, and I hope you all have a good day.